This video will introduce you to Part G, Organic Compounds, the Molecules of Life. All organic compounds are composed of a carbon backbone. Remember, carbon shares a pair of electrons to achieve stability. The shared electrons are shown as a dash between the two carbons. Often, carbon will share the two electrons with another carbon, forming a double bond. Sometimes carbons will share 3 electrons to form a triple bond. A polymer is an organic compound composed of many repeating units. Carbon is special because it can combine with other carbon atoms to form an endless variety of molecules. Glucose 6-phosphate we saw earlier is an example. Very often, ring shapes are common. The carbons are commonly omitted in the ring form, but it's understood that a carbon is present at each angle of the ring. Other atoms are attached to the carbons. Always hydrogen. The compound is a hydrocarbon if no other kind of atom is present. Often nitrogen, oxygen, phosphorus, and others are attached. Complex functional groups may also be attached to the carbons. For example, an amino group, a carboxyl group, phosphate groups. I usually represent the phosphate group with a P and a circle around it. The simplest organic compound is methane. It contains 1 carbon and 4 hydrogens. Examples include a major ingredient of natural gas. It's produced by microorganisms, so it's common in swampy areas, in swamp gas. It's also found in flatulence, and it's been discovered in some of the atmospheres of moons in our solar system. The next simplest is ethane, C2H6, and then propane, C3H8. Organic compounds include 4 main types: carbohydrates, lipids, proteins, and nucleic acids. In describing these, I will list a general function to serve as a frame of reference in understanding the organic compounds. Carbohydrates, the word comes from carbon and water, hydrate, has certain general characteristics. It contains carbon, hydrogen, and oxygen, and the ratio of hydrogen to oxygen is 2:1, as it is in water. It includes roughly 10% of the organic matter in humans. 3 major categories are monosaccharides, literally 1 sugar unit, disaccharides, 2 sugars, and polysaccharides, many sugars. Monosaccharides have as their general function to supply energy. They're classified based on the number of carbons into trioses, which contain 3 carbons, pentoses containing 5 carbons, hexoses containing 6 carbons. The hexoses include glucose, grape sugar, fructose, fruit sugar, galactose, and many, many others. Disaccharides' general function is an intermediate between monosaccharides and polysaccharides. Sucrose is a good example. It's composed of a glucose and a fructose unit. Lactose, milk sugar, is composed of 1 glucose and 1 galactose. Maltose is composed of 2 glucose molecules. Polysaccharides have as their general function energy storage and building material. They include starch, common in plants, glycogen, composed of glucose, as is starch. Glycogen is sometimes referred to as animal starch. It's characteristic of animals. Cellulose, a plant polysaccharide composed of glucose. Among the general characteristics of lipids is they do not mix with water. That is, they are hydrophobic or lipid soluble. They contain carbon, hydrogen, and oxygen, and sometimes phosphorus. Roughly 40% of organic matter in a human is lipids. Many lipids contain fatty acids. If a double bond is present in the fatty acid, it's called unsaturated because the carbons on either side lack hydrogens. Fatty acids without the double bonds between the carbons are saturated. Among the types of lipids are the fats or triglycerides. Fats are composed of 3 fatty acids and a glycerol. They're constructed a little like a long E. The general function is energy storage. A fat is unsaturated if it contains unsaturated fatty acids, saturated if it contains saturated fatty acids. Steroids are another type of lipid. They're composed of 4 rings with side chains attached to the rings. The general function is cellular regulation. A carbon or a nitrogen is understood to be located at the angles of the rings. The names usually contain sterol or sterone or a similar term in them. Yet another type of lipid is the phospholipid. Phospholipids are composed of 2 fatty acids, 1 phosphate group that replaces the 3rd fatty acid. It has a negative charge, plus 1 glycerol. The phosphate group contains other parts attached to it. Here's a simpler view of a phospholipid. The phosphate group represented by the P with a circle around it, the glycerol represented by the straight line, and the 2 fatty acids. We can combine that and simplify it further to look like this. The P with the circle represents, in this case, the phosphate group and the glycerol, and the 2 fatty acids project out to the right. Before continuing, let me remind you that ionic compounds can mix with water, and phosphate groups contain a negative charge. Phospholipids are unique in that they are partly hydrophobic, the lipid-soluble part, and partly hydrophilic, the water-soluble part. This is the simplest view of a phospholipid, and I'll use this model throughout the course. If mixed with water, they form tiny globules with the water-soluble ends facing the water and the lipid-soluble parts facing each other. The general function is membrane building material because if you add more phospholipid to the water, it tends to aggregate into a double layer. It's called a phospholipid bilayer. Another type of lipid is the icosanoid, derived from a 20-carbon fatty acid called arachidonic acid. These include prostaglandins and leukotrienes, both involved in regulation, particularly in the immune system. The next category of organic compounds are the proteins, key components of life. They contain carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur. Roughly 50% of the organic matter in the human body consists of proteins. The number of different proteins is roughly 30,000 per human, perhaps 10,000 per cell. The functions, 2 major functions. One, they serve as building material, and the other, enzymes. Building material includes collagen, a protein that makes up roughly 25% of the total protein in the human. It's composed of 3 polypeptide chains called tropocollagen. Categories of proteins include fibrous proteins. These do not mix with water. globular proteins that do mix with water. Proteins are composed of amino acids linked end to end with bonds called peptide bonds. Types of amino acids are based on the source are essential, which must be present in the diet because they can't be manufactured by cells. They include one called phenylalanine. Non-essential are those amino acids which don't have to be present in the diet because your tissues can manufacture them. It includes, for example, tyrosine. Amino acids contain these parts: an amino group attached to one end of a central carbon, a carboxyl group attached to the other side of the carbon, and the rest of the molecule called an R group. R groups come in a variety of forms, some containing nitrogen or sulfur, some containing positive and negative charges. Roughly 20 different amino acids exist. What makes them different are the R groups. Some contain just hydrogen, some are charged positively, some have a negative charge, some contain nitrogen, others contain oxygen, and still others contain sulfur. The number of amino acids determine the molecule. If two amino acids are linked, it's called a dipeptide. If you link three together, it's a tripeptide. More than three would form a polypeptide. If you have more than 100, we generally refer to it as a protein. But there's really no difference between a large polypeptide and a small protein. Peptide bonds are a type of covalent bond. By the way, how many peptide bonds does a dipeptide have? Be careful. You probably said 2 because of the di. I was just trying to see if you're paying attention. It has one peptide bond, 2 amino acids joined by one bond. Peptide bond links the carbon of one amino acid to the nitrogen of another. Remember, to become stable, carbon forms 4 bonds. Nitrogen, to be stable, has to have 3 bonds. To form a bond between the carbon and the nitrogen, a bond must be broken on each to free up an electron to share. The carbon loses an OH, and the nitrogen loses a hydrogen. The OH and H combine to form water. The reaction is dehydration, or dehydration synthesis, the removal of water to link two molecules. To split the bond, the reverse happens. Water splits into hydrogen and OH. The hydrogen is added to the nitrogen to make it stable. The OH is added to the carbon to make it stable. The reaction is hydrolysis, that is, the addition of water to split a bond. Another name for hydrolysis is digestion. Now, notice that dehydration and hydrolysis occur among all organic compounds. I'm just using the amino acids as an example. The shape of a protein is critically important for its function. Protein shapes exist in several levels. The primary structure is the straight chain due to the amino acid sequence linked by peptide bonds. Higher levels are due to the interaction of ionic bonds, hydrogen bonds, and sulfur-sulfur bonds in the R groups. The secondary structure is a coiling or a folding of the chain into an alpha helix or a beta pleated sheet. The tertiary structure, the third level, is the overall shape. Weak bonds result from the interaction of the R groups, causing kinks to form in the chain. Positive and negative charge attract one another, causing a bend. Sulfur-sulfur forms bonds, causing another bend. And hydrogen and oxygen form hydrogen bonds, causing another bend in the structure. The quaternary structure of a protein is the interaction of two or more polypeptide chains. Notice they're not linked end to end. They're simply combined into the overall structure. To denature a protein is to disrupt the secondary, tertiary, or quaternary structure of the protein. Heat, acids, and bases will denature a protein. Here, for example, is a protein with its overall shape. When we denature it, these weak bonds have been disrupted and the protein is laid out in a straight chain. A good example is egg white, albumin. In the fresh egg white is a gooey, semi-liquid, semi-transparent material. You put it in a hot frying pan, and what happens? Quickly, these weak bonds are ruptured, the chain of amino acids suddenly straightens out, and you have straight chains of albumin in the frying pan. It has become white and solid, and you have ruined it for the chicken. But has this changed the nutritional quality of the protein? Not at all. It still consists of the amino acids it originally had. Protein metabolism involves two things: the manufacture of proteins, called protein synthesis, and the breakdown of proteins, called protein degradation. These are continuous processes going on in a cell. On the average, a protein may last a few hours to many weeks in a cell. Like most of the components of a cell, the proteins are constantly being recycled. Many proteins exist in an inactive form and have to be activated to work. For example, fibrinogen is activated to fibrin, pepsinogen activated to pepsin, and prothrombin activated to thrombin. Enzymes are organic catalysts. They increase the rate of a reaction. For all practical purposes, that means if the enzyme is absent, the reaction will not take place. They're very specific. Each different reaction needs a different enzyme. So if a cell has 10,000 different reactions, there will be 10,000 different enzymes required. One enzyme may catalyze many millions of reactions in a minute. This may be 10 million times faster than without the enzyme. Enzymes are located, fixed, attached to cell membranes, in solution in the cell cytoplasm, or secreted from the cell. Enzyme names usually end in A's. Example of enzymes include protease that splits proteins, lipase that splits lipids, kinase that transfers phosphate group. Other organic catalysts exist, including RNA molecules called ribozymes, which split RNA. The substrate is the molecule the enzyme acts on. Here are two molecules that combine to form a product. Each molecule is the substrate of the enzyme: substrate A, substrate B, and substrate AB. Enzymes contain an active site, which is complementary to the substrate molecules. To function, the enzyme binds specifically to the substrate. The substrate molecules remain bound long enough for the reaction to occur, and then the enzyme releases the substrate and immediately catalyzes a second reaction. If the enzyme is denatured, that is, its shape is changed, it will no longer work. For example, if the pH changes, this might result. Let's say this is an enzyme with a certain shape. If the pH changes ever so slightly, it might alter these positive charges and change them into negative charges. If that happens, these two sides of the active site are pulled closer together. When that happens, the substrate molecules no longer fit and the reaction will not occur. The fourth major group of organic compounds are the nucleic acids. Nucleic acids contain carbon, hydrogen, oxygen, nitrogen, as well as phosphate. They have two important functions. One is to store genetic information, and the other is to make proteins. We call it protein synthesis. There are two types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. There are three major forms of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Nucleic acids are composed of nucleotides. Each nucleotide contains a 5-carbon sugar, a phosphate group, and a nitrogenous base, a compound containing nitrogen that has basic characteristics. The nucleotides are joined by the phosphate groups. For simplicity, the phosphate groups and the sugars may be represented by a straight line. Two types of 5-carbon sugars are ribose, found in RNA, and deoxyribose in DNA. Two groups of nitrogenous bases are purines with a double ring structure. They include adenine and guanine, and the pyrimidines with a single ring structure, including cytosine, thymine, and uracil. Note that thymine is found only in DNA, uracil is found only in RNA, otherwise they're rather similar. Strands of nucleotides exist. Messenger RNA is generally a single-stranded molecule. Transfer RNA is a single strand folded in an irregular shape. In this diagram, only 3 of the bases are shown. Ribosomal RNA forms a complex structure tied together with protein. DNA is a 2-strand nucleic acid. The strands are side by side by pairing complementary bases. For example, adenine is complementary to thymine. Cytosine is complementary to guanine. The paired strands are coiled into a double helix. It's as though you took a ladder, grasped it at both ends, and twisted it. Note the importance of the hydrogen bonds between the bases. Between thymine and adenine, the distances are just right for 2 hydrogen bonds. Between cytosine and guanine, the distances are just right for 3 hydrogen bonds. One other organic compound needs to be introduced, and this is ATP. It's a major form of energy used by cells, so it's a primary energy carrier. ATP is composed of adenosine, which is like adenine with the sugar, and 3 phosphate groups. When the third phosphate group is removed, energy is made available for use by the cell. The enzyme to split ATP is ATPase. Where does the ATP come from? It's generated mainly by cellular respiration. We'll cover this in the next unit. The enzyme to form ATP is ATP synthase. Organic compounds can be combined in various ways. Glycolipid, for example, is composed of carbohydrate and lipid. Glycoprotein, carbohydrate and protein. A lipopolysaccharide is made of lipid and carbohydrates. Lipoproteins are composed of lipids, and proteins. And that is a whirlwind look at matter. You're now ready to continue to the next unit, Unit 3, Cells.